Okay, so we got 18 people. This one's coming in at 18. Okay, so first of all, in the chat box, would you please let me know what 2 phases of the cell cycle you see in this image at the top? In the chat box, 2 phases that you see. Looking at mitosis. Gabby's on track. Elizabeth is on track. Riley's got it. Okay, everybody's coming in pretty good. So metaphase, they're lined up at the equator, right? Meta, middle. Chromosomes are lined up at the equator. Ana, apart. Chromosomes are being pulled apart. So we're going to hit some of our last topics this week. We're going to cover stem cells today. Originally, I was going to do all 3 topics today. They're all very short. But I decided I took up enough of 2nd hour's time, or whatever's class, 4th hour's time. So I'm going to save the apoptosis and cancer for Thursday. Today, we're just going to hit the stem cells. So we'll play a Kahoot to practice mitosis. We'll talk about stem cells, and then on your own, you're going to do the stem cell web quest, where you're going to cover this topic in much more detail than I'm going to do in class. And then you want to work on just that chapter 1 if you haven't already done it, so that I can field questions over chapter 1. Right? So let's go ahead and get the Kahoot started. So if you didn't get in, just answer them on your own on the side, okay? So you can still practice recall this way. I'm looking at this cell and I see 2 nuclei. 2 nucleus means it's in the process of separating. That would be telophase. Bella takes a quick lead. She's already looking at that 1 point extra credit for the day. Bonus points. Organelle in animal cells, not in plant cells. Dylan, we started a game of Kahoot. I don't know if you can join late. If not, I'll throw the code in here. If you can't join late, then just maybe do the answers on the side to practice, or I put the link in the Google Classroom. You could do an independent game. Anybody who didn't get in could do an independent game. So we identified some differences in cytokinesis between animal and plant cells. Animal cells just pinch in. What do plant cells have to do? Animal cells or plant cells have to form a cell wall, right? Which comes from the cell plate. Kind of like an extra membrane. I can see where you got that. Jonathan takes the lead. Miranda holds steady in that number 2 slot. What actually splits the cytoplasm of the cell? Pinches it in half. Splitting it in half, cyto means cell, right? Cytoplasm is popped into the top 3. When do we see the chromosomes? When do they become visible? When do the spindle fibers form? So this has to be somewhere at the beginning, right? Prophase. Spindle fibers form. You want to know that for the test. Abby's on a streak. Gabby takes the lead. Nuclear envelope reforms. It went away at the beginning. It comes back at the end. When does it come back? It comes back during telophase. Bella's holding strong at the top 5. Somebody new is coming into play. Okay, they don't seem like they're quite at the center line. So it's going to be shy of the center. Prophase it is. It almost looked like metaphase. Metaphase, they got to be right in the middle. Sartage makes a big jump. Nice work. Gabby's holding strong at the top. Do you make 2 identical cells during mitosis? Or are they different? They are identical. You need to know that for the test. Same contents inside, same genetic information, same number of chromosomes. Ooh, no movement on the board, but Riley is on a streak. We've seen this one already once today. They're lined up right down the midline. You're going to see a picture like this probably on the final exam. That's going to be metaphase, meta middle. PMAT, meta middle. Gabby is on a streak. She's got 7 in a row. Bella's making moves. Top 3 are going to get a bonus point today. This is guaranteed test question. Given an original number, how many will the daughter cell have? You have 24 to begin with. You go through DNA replication, make copies of each of those, and then you split in half. So each cell gets a copy, 24. Otherwise, they wouldn't be identical. Abby is on a firing streak. She's in the number 2 spot. Gabby is holding tight to number 1. She is not giving it up. The prefix inter literally means in between. Interphase is between the phases. Used to be thought of as a time of rest, but now we know lots is happening. Jonathan's coming up, challenging the top 3 spots. This should be the last question. You can't get it wrong. You feel like you're going to ace the mitosis portion of the test. You feel like you kind of know it, but you could definitely use a little more study. I'm going to be at the NHS tutor table today if you want to join at 2:30 if you need any extra help. Sophia is on a streak. Centromeres, the point where 2 chromosomes are attached. Oh, that. When are they lined up across the center? Lots of different ways of asking the same question, basically. Right here, they are lined up across the center. What phase is that? Metaphase. Anna apart, meta middle. A little bit of movement. Sophia's in the top 3. Elizabeth has made some big jumps. Last question, who's the top 3 going to be? Get my pen and paper ready. Chromosomes separate. They move apart. Anaphase. Apart. Okay, so our top 3, Gabby, Sophia, and Abby are going to get bonus points today. Nice work. If you want to practice on your own later, we're under special topics today, week 18. So I have this Kahoot game you can play independently if you'd like. Tonight we're going to do some stem cell web questing, or today during our flex time, you'll have an hour and a half to do that. And then we're going to just talk very briefly about stem cells today. All right, so over the weekend you were charged with doing chapter 9 on the exam review packet, and that I would field questions related to chapter 9. So in the chat box, or shout it out, either way. Our exam review is in the 19th week. You can open that up. I think we jumped all the way down to like page 10. Any questions I can help you with? Because I'm not going to go through the whole thing. I'm not going to go and give you an answer key because this is really your test, not mine. So we start on page 9. Did you have any questions on chapter 9, pages 9, 10, and 11? If you don't know something on the review, rather than leave it blank, ask me right now so I can help you fill it in. So go over G1, synthesis, and G2. So I'm up here. So during the G1 phase, that's cell growth, right? So making sure that the cell is large enough to support two separate cells. Also going to make copies of each of your organelles. So one cell has to become two, so you need enough nutrients, which you'll find in the cytoplasm, and enough organelles to support two separate cells. The S phase, I guess that doesn't describe it. The S phase is when DNA is replicated, synthesis of DNA or DNA replication, which again is important because one cell needs to become two cells. And if one cell has all the genetic information, both daughter cells need copies. But if you don't make a copy of your genetic material, when you split in half, each daughter cell only gets part of it rather than a copy of each. So S phase is important to make a copy of each of the genetic materials. And then the G2 phase, this is when we start to assemble spindle fibers and materials that we're going to need for the division itself. So many of your organelles, as well as those protein fibers that we'll use to split the cell, are going to be made during the G2 phase. So after we're done with Zoom, I'll go through and I'll add this to our agenda slide in case you don't get a chance to get all that information down. Okay, Miranda, is there one I can help you with from chapter 9? Okay. So each day, Miranda, I'm going to ask you guys for questions on a chapter. So if there's something you don't know from one of the chapters, and this is true for everybody, right? Only Miranda was brave enough to ask. So rather than leave it blank, make sure you ask that question when we field that day. Can I go over number 2? Isabel, I absolutely can. Because you know what? That was a question already today. So the question was, in number 2, you're comparing or differentiating between sisters, which are identical copies, and homologous chromosomes, which are similar but not identical. Okay, so sister chromatids, remember we start with 46 chromosomes and then we make copies of each of those. So the sister is the copy. So here I have, you see when they're in X's, these are sisters. So it's a relationship between the two. It's the copy that's made during the synthesis phase. Homologous is similar. So mom's version of number 1 is homologous to dad's version of number 1. They're not identical. They might, like, they both carry information for hair color, eye color. I'm just making that up, but let's assume they both carry information for hair color and eye color, but they might not carry the same information. Maybe mom says blue eyes and blonde hair, and dad says brown eyes, brown hair. So similar in the information that they carry, same chromosome length, centromere position, but not identical. Now I'm going to go to the definition page so I can maybe give you a better picture of this. Yeah, all these boxes that I don't really want. So the greens are identical, right? So they're sisters. So here we have mom's number 5 and dad's number 5. During the S phase, we make copies of mom's number 5. That copy is its sister. So the 2 greens are sisters with each other. The 2 pinks are sisters with each other. The green might be mom's and the pink might be dad's. So they carry information for the same traits, whether or not you will have lactose intolerance, for example. Moms might say yes, dads might say no. So they're not identical, but they carry information for the same traits. If that makes sense? No worries, Kylie. Welcome, welcome, welcome. Okay, so in 4th hour, they also asked about the definitions, which kind of is a repetition of that number 2, where I asked you to differentiate between the 2 You also can identify centromeres. So sister chromatids are identical. Homologs have their centromeres in the same position. So the centromere is the point where two sisters connect. Okay. Are there any other questions from chapter 9 that I can field? Number 5. Okay. I have to exit out because those were the two I've done so far. Number 5. What is the role of the spindle fibers? So their job is to move chromosomes around during mitosis. So they make sure they align at the equator and that they pull apart. So just because I like to have images when I'm talking spindle fibers. So these strands, right? They arrange the chromosomes along the equator and then separate them. So their job is simply to move the chromosomes during mitosis. After mitosis, the parent cell can say, okay, always going to be the same number. So no matter what number you're given, after mitosis, you'll have the identical number. So we have 46 chromosomes. After our cells separate, we have 46 chromosomes in each daughter cell because you made a copy during that synthesis phase, right? So in this example, you have 12 chromosomes. Each daughter cell will have 12. If they had 48, each daughter cell would have 48. If they had 72, each daughter cell would have 72. So the number is the same, okay, no matter what. Stem cells is what we're going to cover today, actually. And basically, they are unspecialized. So I'm going to maybe jump over to the PowerPoint at this point because that's what we're going to talk about right now. So they're unspecialized and they're used basically for medicine and research. Okay, so we're going to work on chapter 1 later today. I'll ask questions about chapter 1. Tutoring, I put a link in. They meet every Tuesday and Thursday after school for help. And there's always a teacher in the room as well. And I'm that teacher today. So if you wanted more help with any of this, you can come to that tutoring. I gave you the link and you can come in and get help from me, or if you want help for a different class, you can do that. So today's special topic is stem cells. And you can go into a great amount of detail. I am not going to do that. You are going to cover this in more detail when you do your web quest. That's the word, web quest. Today we're just going to kind of talk about what stem cells are and what they're used for. So we'll start out with a relatively short video. And they do a nice job explaining this or summarizing it, basically.
Are cells that are undifferentiated, meaning they do not have a specific job or function. While skin cells protect your body, muscle cells contract, and nerve cells send signals, stem cells do not have any specific structures or functions. Stem cells do have the potential to become all other kinds of cells in your body. Your body uses stem cells to replace worn-out cells when they die. For example, you completely replace the lining of your intestines every 4 days. Stem cells beneath the lining of your intestines replace these cells as they wear out. Scientists hope that stem cells could be used to create a very special kind of personalized medicine in which we could replace your own body parts with, well, your own body parts. Stem cell researchers are working hard to find ways in which to use stem cells to create new tissue to replace the parts of organs that are damaged by injury or disease. Using stem cells to replace damaged bodily tissue is called regenerative medicine. For example, scientists currently use stem cells to treat patients with blood diseases such as leukemia. Leukemia is a form of cancer that affects your bone marrow. Bone marrow is the spongy tissue inside your bones where your blood cells are created. In leukemia, some of the cells inside your bone marrow grow uncontrollably, crowding out the healthy stem cells that form your blood cells. Some leukemia patients can receive a stem cell transplant. These new stem cells will create the blood cells needed by the patient's body. There are actually multiple kinds of stem cells that scientists can use for medical treatments and research. Adult stem cells or tissue-specific stem cells are found in small numbers in most of your body's tissues. Tissue-specific stem cells replace the existing cells in your organs as they wear out and die. Embryonic stem cells are created from leftover embryos that are willingly donated by patients from fertility clinics. Unlike tissue-specific stem cells, embryonic stem cells are pluripotent. This means that they can be grown into any kind of tissue in the body. A third kind of stem cells is called induced pluripotent stem cells. These are regular skin, fat, liver, or other cells that scientists have changed to behave like embryonic stem cells. Like embryonic stem cells, they too can become any kind of cell in the body.
These, that last set, those are pretty much the newest in the research, where you can take a skin cell, and once it's a skin cell, it's really just ever going to be a skin cell, right? It's committed to that job. But that last one that he was talking about, they actually can reverse the genetic coding and cause it to like turn back time and become a stem cell all over again.
Are cells that are undifferentiated.
Sorry about that. Okay, so basically, and this is your last question on the exam review packet, what is special about stem cells? They have the ability to renew or make copies of themselves, so they can make more stem cells that can become anything. They're undifferentiated, meaning they don't yet have a specific job, and so they can still do any type of job. Like you start out as a single egg, right? And it's fertilized by a sperm. Well, how does that egg become a skin, an eye, a liver? All of this tissue in our body is so very different, right? That's cell specialization. So it's changing that cell into something new. And only a stem cell can do that. Once a muscle cell becomes a muscle cell, it can only ever be a muscle cell. Once a bone cell becomes a bone cell, it can only ever become a bone cell. But you have, like, in your blood, in your bone marrow, right, inside of your bones, you have hemocytoblasts, which are they're blood cell stem cells. So they can become any type of white blood cells. You have a handful of different types of white blood cells that fight infections. You have platelets that clot blood. You have your red blood cells that carry oxygen. None of those cells will replicate, and so none of those cells can become new white blood cells, red blood cells, or platelets. But that hemocytoblast can still become any one of those types of blood cells. So it's somewhat differentiated, but not all the way differentiated. That's the difference between a totipotent and a pluripotent. And you'll read more about that. So these stem cells, given the right signals to turn on genes, can become nerve cells, they can become endothelial cells, they can become liver cells, heart cells, bone cells, any kind of cells. I have lack of cartilage on the bottom of my knee. Maybe Abby's sister can relate. And so what they did, I had surgery and they drilled into my bone. So on the bottom of my femur bone, that's where your knee joint is, right? So they drilled into the bone to release the bone marrow stem cells that then should fuse with my cartilage and become new cartilage cells. How crazy is that, right? So that is differentiation. So they're undifferentiated inside my bone marrow. They could become any type of cell. They could become cartilage, they could become bone, they could become blood. Then released, they'll start to get signals from surrounding cells as to what their job is. And that turns on certain genes that then commits them to a certain job. Okay, so stem cells, what's special about them? They can be used they can become any type of cell. What are they used for? As we saw in that very short video, They can be used to treat disease. Like if you have diabetes, your pancreatic cells don't make the gene or the cells that you need to make insulin. You can use stem cells to recreate those cells so that person can make insulin. Regenerate tissue, like I discussed in my knee surgery example. Or let's say you're a burn victim and you've been burned like 80% of your body. There's not a lot of skin available for you to make skin grafts with, right? And you may reject skin from some other person because your genetic codes don't match. They can actually take your stem cells and grow your skin in the lab so that you can then have a skin graft to cover up that burn. They can even 3D print your skin using those stem cells to produce like the cells that they use as the material that they put in the printer. That's insane. That's so awesome. Okay, we also use it in research to better understand those diseases and to better understand the whole process of tissue development. Okay, so 2 things you needed to know about stem cells. What's special? They're undifferentiated and can become any type of cell. What are they used for? Treat disease, diagnose disease, or not diagnose disease, treat disease and tissue repair, and then research. Okay, so that should answer that last question from chapter 9. We're going to take a break there. You have a stem cell web quest to work on. You're going to get much more information on stem cells. And then we'll do our last topic, which is super short on Thursday, apoptosis and cancer. Okay.